Good afternoon, everyone. I hope you had a satisfying lunch and a cup of coffee. And I'm really happy you've chosen to join us for this special session. I have the great privilege of introducing two past presidents of the Genetics Society of America, who are going to lead a discussion with a truly impressive panel of experts on a topic of critical importance for our field. Tracy Johnson is the Keith and Cecilia Terasaki Presidential Endowed Chair in the Life Sciences, Professor of Molecular Cell and Developmental Biology, and Dean of Life Sciences at UCLA, as well as an HHMI professor. She was president of GSA last year and is currently serving as an immediate past president. Tracy's tenure at UCLA has been marked by her dedication to advancing diversity, equity, and inclusion, and we're grateful that she brought that dedication to her work at GSA. Denise Montell is a Dugan professor and distinguished professor of molecular, cellular, and developmental biology at UC Santa Barbara. She served as the GSA president in 2020, presiding over our pandemic TAGC, and deftly leading the society through the uncertainty of that year, including helping to found the Presidential Membership Initiative. Denise and Tracy convened this panel to discuss eugenics, the roles it played in the early days of our field, the ways it creeps into our current lives, and what we can learn to carry us into a more just scientific future. We can't wait to hear this discussion, so with that, I'll turn it over to Tracy and Denise to introduce our panelists and kick things off.
Thank you so much for that introduction. Good afternoon, everyone, and welcome to the discussion, the eugenics movement and scientific racism, past, present, and future. I think I speak for both Denise and myself, both past presidents of GSA, when I say that we were drawn to leadership of this society, not only because of the extraordinary research that you all do and the ability to advance the work that's happening among geneticists, but also because the GSA understands that scientific research takes place in the context of society, and that this organization has the goal of building an environment in which all researchers from all backgrounds are welcomed and thrive. And so we recognize that given that there's a larger context of the science we do, and given the kind of environment we want to create, we have to look critically at the space in which we do our research. And so this is a critical time to be reflective and honest about our past, which is the genesis of our discussion today. So for context, this conversation happens in an environment that is more broadly really having to grapple with some challenging issues. One need only look at examples from recent publications in which questions are raised about the way data is presented about populations and how it visually represents what the concept of race means. And there are growing trends, of course, of problematic and non-scientific organizations who are using genetics research to support racist ideology. So we felt that it was critical at this moment to engage as a community in a conversation about eugenics. So I want to start by acknowledging a few points. First, we are not going to be oversimplifying the topic because it's complex. Secondly, as genetic scientists, nuance and complexity should be very familiar ground for us. So we're going to start with that assumption as well. We do recognize, nonetheless, that many people will experience this topic differently and hope that you'll be able to sit with sometimes the discomfort that that leads to. We may be talking about individuals who are luminaries in genetics, people who have made profound impact on the research that we all do, and that is not being denied at the same time that we take the opportunity to reflect on some of the context in which the science they were doing lies. And finally, it's important to say that we entered this conversation from the perspective of rejecting eugenics ideology while being fully cognizant of the need to commit to understanding how eugenics ideology have lived within and outside of our institutions, as well as our societies. So now I am pleased to introduce our fantastic panel of experts. Nathaniel Comfort is a professor of the history of medicine at Johns Hopkins University, who studies the histories of heredity from late 18th century to the present. Special interests include interests in genetics, eugenics, molecular biology, and genomics. He's widely viewed for his scholarly work and his interactions and engagement with lay audiences on the hazards of genetic determinism. Nathaniel Comfort is part of the Hastings Center Working Group wrestling with the risks and benefits of genomic studies of complex social behavior. Our second panelist is Stephen Farber, professor in the Department of Biology and director of the Cell, Molecular, and Developmental Biology and Biophysics graduate program at Johns Hopkins University. He uses zebrafish larvae to study the molecular and cell biology of lipids in live cells and organs. He's the co-creator of BioEyes, an outreach program using zebrafish to promote science literacy and teach genetics and experimental processes. The goal is to instill a love of science to all students and teachers, regardless of community, income, or race. He's published the important work, U.S. Scientists' Role in the Eugenics Movement, 1907 to 1939, A Contemporary Biologist's Perspective. Our next panelist is Daniel Martinez HoSang, who's a professor of American Studies at Yale University and holds secondary appointments in the Yale School of Medicine section of the History of Medicine and Department of Political Science. He's the author or co-author of 6 books on racism and racial justice and conducts research on far-right political movements in the United States. He leads a collaborative research project on the history and afterlives of eugenics research and advocacy. And in this context, he conducts educational workshops with colleagues in medicine, in sciences, as well as in the K-12 environment to really examine histories of eugenics in institutional contexts. Jazlyn Mooney is the Gabelin Assistant Professor in the Department of Quantitative and Computational Biology at the University of Southern California. She uses whole genome sequence data and computational methods to understand the evolutionary and population histories of both human and non-human populations. She's a population geneticist with training in anthropology and human genetics. And Dr. Mooney has taught and teaches courses on the ties between genetics and eugenics. And finally, one of our panelists who is joining us remotely, Katrina Claw, who's an assistant professor of biomedical informatics at the University of Colorado Anschutz Medical Campus. She uses genetic information and biomarkers for tailored treatment of populations historically underrepresented in health research, including studies of cytochrome P450 genetic variation in indigenous communities. The work that Dr. Claw does is really driven by community-based participatory research approaches. She served on the National Academy's Committee on the Use of Race, Ethnicity, and Ancestry as population descriptors in genetics research. Dr. Claw grew up on Navajo Nation and is an enrolled member of the Navajo Dene tribe. And so with that, I'm going to turn it over to the panelists. Each will have about 5 minutes to lay the groundwork for our discussion. Then we'll have moderated question and answer, and then we will open it up for an opportunity for discussion and questions from the audience. So we'll start first with Dr. Comfort.
Well, thank you, Tracy and Denise. It's a really wonderful opportunity for me to be here. I'm thrilled to attend this meeting and that I've studied as a historical institution for many years. So it's my job to introduce the idea of eugenics and try to toss out a couple of provocative points. First, I'm going to start with where the term came from. The term eugenics was coined by Francis Galton, who was a half cousin of Charles Darwin, and polymathic, interested in statistics and so forth. And he got the idea that now that Darwin had explained how heredity works, we should be applying it to human betterment, right? To make the human race better. It's had a very colonialist flavor, but it was also influenced by the kind of Dickensian conditions in 19th century London, the New Poor Laws and so forth. And so these issues of class and colonialism were really motivating for Galton. And that's where his idea of eugenics came from. But he did not believe in compulsory sterilization until the very end of his life, when he was persuaded of it by his very right-wing protege, Karl Pearson. So, the English really never had much appetite for compulsory sterilization. The rediscovery of Mendel's laws in 1900 needs to be seen in this context, I think. More important even than Gregor Mendel himself is William Bateson, an English naturalist. He translated Mendel's work into English for the first time and gave us much of the language of classical genetics, heterozygote and homozygote and many other terms, that alleles, so forth, all were Bateson's coinages. So he's a very formative figure in the history of genetics. Like Mendel, Bateson treated traits as binary units. He thought it was an element that was either present or absent. That was dominant and recessive for him. Like Galton, he treated heredity and environment as distinct and separable. And he laid great emphasis, and this is the key point, on the purity of the gametes, gametic purity. And that's very much tied to Mendel's own experiments, which he bred his famous crosses of peas from pure breeding lines. And Bateson brought that idea of gametic purity forward. Very important for the social history of eugenics. American eugenics in the progressive era, from the discovery of Mendel for the first couple of decades, was really where eugenics took off as a broad-based scientific and social movement. And it developed in the context of the progressive era, which is a time of great appetite among the entire population, the whole spectrum, for ideas of social reform. How do I go forward? Oh, it is doing, okay. And are those coming up? All right, where are they? My animations aren't working. I'm sorry. Let's see. Okay, there we go. Characteristics of the progressive era include a very strong spirit of reform of all sorts, from health to politics to birth control, a faith in government as the most powerful tool we had to effect that social reform. Not the only, but the only group that was powerful enough to stand up to the robber barons who had, in the 19th century, who had created all these problems in the first place. And third, scientism. And by scientism, I mean not just science, but the philosophical belief that science is the only way to describe or solve or address any kind of problem, including social problems. Okay. And the results of this, of progressive era American eugenics, were a variety of laws because of that faith in government. So these reforms took the form of things like marriage laws, saying who could marry whom, both with racial restrictions, but also disease restrictions and various kinds of things. Immigration laws. It was seen as a very big social problem by many of these people, that this was a time of great immigration to the United States from Europe on the East Coast and from Asia on the West Coast. And many people, as always, felt threatened by that. And finally, eugenic sterilization laws, which really are part of this whole idea, the idea that the state has a responsibility to improve the overall heredity of the American population. Okay, so it's in that context that Charles Davenport created the Eugenics Record Office at Cold Spring Harbor in 1910. Davenport was a Harvard-trained zoologist, one of the first Mendelians in the United States, and very interested in using this new knowledge of heredity. Now that we understood heredity as a Mendelian thing, we should be using it for social improvement. And the Eugenics Record Office was his main vehicle for doing this. Davenport was director. This was the third institute in Davenport's little empire of institutes at Cold Spring Harbor, New York. It is now united into Cold Spring Harbor Laboratory. Okay? And so he defined eugenics as the science of human improvement by better breeding. So from Galton to Davenport, we see this hardening of eugenics into a coercive, government-run, state, top-down idea, focusing on specific people being allowed to breed or not. Okay, and that's all I have to say. Thank you.
So I'll pick up with Davenport and just say, like, what I do every day is, and I think what most of us in this audience are doing, is we're about knowledge creation. So we're not actually doing that much retrospective analysis. We just look at the history a few years back, or to know that we're looking most carefully at a particular knowledge gap. And we take that historical information in our specific fields and we push forward into the unknown and we try to create new knowledge. And that's what we're doing every day. But today we're going back 100 years, more than 100 years. And I think the reason we're doing this is to understand better that climate, and to know, in this case, you'll see there were luminaries of science, Nobel laureates of science, that created the systems, the model organisms that we use. And there was a conflation between science and folklore, if you will. And I want to put out there that it's easy to look back as Monday morning quarterback to know where that line is. But the question we all have to ask ourselves is, if someone were to look back 50 years from now, they could look at our work and say, have we gone awry? And that's the question we're grappling with, I think. So having just submitted my NIH revision for the Tuesday deadline, Davenport wrote a grant to the Carnegie Institution. It was $45,000. Carnegie, by the way, was my former institution. So I had all these books in the library at Carnegie to explore some of the history of this. And it would be about $1.6 million in today's terms. And it was only 2 pages. And in that grant, if you notice, the aim that really motivated Davenport was the experimental study of the causes of race change, if you will. And he was definitely motivated by the breeding stock of people. But the brilliance, if we will, now we're not lionizing Davenport for his eugenics, but there's a brilliant thought he had, or what he pushed forward, was we need to have model systems. We need to collect different animals, different plants, and then we could study inheritance. And when we look back, that was a transformative idea. And I think this is the first place where we see the execution of the idea we put in the brilliance category, the motivation. We now look back and we say, huh, you know, what did that lead to, let's say. Next slide, please. So in Morgan at that point was aware of Davenport's work, and Morgan saw the power of the Drosophila system, so that by 1921, Sturtevant and others at Columbia were getting support from Carnegie to kind of set up the Drosophila system. And as some of you might see from one of my postdocs this week talking about positional cloning of mutants, it blows my mind how they figured out and assembled the chromosome when they didn't really even understand what a chromosome was. But here's the 1921 basic map of the classical Drosophila mutants, and they figured out so much. So that is truly a milestone in genetics. And so you see how Morgan fits beautifully with the system that Davenport was trying to do. And if you look back to, next slide, please. If you look back to, let's say, by the 1920s, and every year in the Carnegie Yearbook, it will list the summary of accomplishments. And I cut the page off at the bottom, but there's 12 things. And right at the number 1 was Morgan's work at Columbia. Down in number 8 is the analysis of juvenile traits and hereditary characteristics of successful naval men. So here we have love of the sea being studied as a sex-linked trait because it wasn't seen in females. And it was also related to wanderlust. And that is right there in the report as number 8 with the whole establishing of key aspects of Drosophila chromosomes. So that's kind of sets the stage for what was going on in 1920. Next slide. So I also want to mention in 1921, there was the Second Congress of Eugenics. And at this time, Davenport was separating the genetics office and genetics studies at Cold Spring Harbor for his new eugenics effort, which was the records office. And he was getting support from Carnegie, from AAAS, from the American Breeders Association, and other powerful entities, including John D. Rockefeller, the Kellogg cereal person, and others, because of pushing forward his idea of racial purity and the need for us as a society to maintain the breeding stock using these ideas of Mendelian heredity. And this, I wanted to point out that this was not a fringe idea. This is the cover story in 1921 in Science Magazine. If you go to the next slide. And in the United States, this is a quote from that first page. In the United States, we are slowly waking to the consciousness that education and environment do not fundamentally alter racial values. We are engaged in a serious struggle to maintain our historic Republican institutions through barring the entrance of those who are unfit to share the duties and responsibilities of our well-founded government. The true spirit of American democracy, that all men are born and with equal rights and duties, has been confused with the political sophistry that all men are born with equal character and ability to govern themselves and others, and with the educational sophistry that also that education and environment will offset the handicap of heredity. So we see this conflation, this driving of the genetic work into real policy that led to forced sterilization. Many of these scientists, Davenport including, served on the racial hygiene journals in Germany. So we cannot hang the final solution on essentially killing all these people that were deemed unfit. We cannot hang that on American eugenicists. But I can say, and many historians can say, that this was the mainstream idea that the Nazis took completely from this rich literature in the 1920s and 30s. And the last part I'll mention is, you know, we're always struggling about some of our leaders. I brought up Morgan because, you know, we're deciding, can we give out a Morgan Award or we can't? Was he a eugenicist? Was he not a eugenicist? And I think that in talking to Nathaniel, he mentioned to us, the importance of these ideas about like traits being unidimensional, like one being single Mendelian traits, binary, right? And Morgan is struggling with this. So, and people claimed at the time, like, or historians after this were looking back and say, why didn't Morgan fight back? You know, why didn't he challenge Davenport? And this quote, I think, shows you where he was at. And he attacks Davenport and Langland. He doesn't name their names, but everyone knows what he's talking about. The case most often quoted is feeble-mindedness. That has been said to be inherited as a Mendelian recession. But until some more satisfactory definition can be given as to where feeble-mindedness begins and ends, and until it has been determined how many and what internal physical defects may produce the general condition of this sort, and until it has been determined to what extent feeble-minded is due to syphilis, it is extravagant to pretend to claim there is a single Mendelian factor for the condition. And until all the social conditions surrounding the childhood of the individual are examined and given proper weight, seriousness, doubts will arise as to what form of inheritance is producing the results. So I think I wanted to share that quote because if Morgan thought it was a single gene, maybe he would have been all in as a eugenicist, but the data was not supporting that for him. So as we reevaluate this kind of history, we have to kind of try to imagine where would we be during this moment. And that's all for my intro.
All right. Hi, everybody. Thanks so much, Tracy and Denise, for bringing us together in this way. Pleasure to be here. Not a pleasure to talk about eugenics, but a pleasure to be here. I want to do three quick things in my opening time. Tell just a short story that's in the present about kind of how your world, the world of genetics research, came into my world, the kind of historical study of race and racism. Talk a little bit about the paradigms that we often use to teach about how scientific knowledge is reproduced, and then share some material examples of how we engage colleagues at my campus about the very, very specific history of eugenics research there. So just a quick story. 15 years ago, I go to my department's colloquium. A colleague in sociology comes and says, I've been reading intensely in this new work in population genetics. I have a new theory that they've disabused the idea that race is a social construction. In fact, they've discovered it has a biological basis. This is 2008. And that biological basis might explain group-based differences, intelligence, athletic ability. It might explain ethnic conflict. There was an automatic deference from many of us in the room, social scientists and humanists, that said, well, if someone in a white lab coat said it and you have some tables, some of it must be true. I spent the better part of 2 years reading Rosenberg and other, colleagues in your fields who offered very careful cautions. Don't use the work for this. But it was startling to watch how quickly, it was like a game of white supremacist telephone. Starts in the papers, a couple of other people pick it up. By the time the piece came out, white supremacist websites within 2 days put it up and circulated it. See, we told you all along, it's true. Now we might take this as a case of, and then I had to scramble with other colleagues to learn it and to try to offer, a critique. It's a case of the misuse, right? We have to simply speak up. But I want to suggest that there's something about the structure in the ways that we teach about scientific knowledge production that leaves fields open to these kinds of distortions and pressures. So just behind me here is one sense, and apologies for a certain kind of simplification, but in many fields, this is how we teach about knowledge discovery. We might say we take the scientists, the researchers, we lock them in their room and imagine that all of it just took place there. So we start with Mendel here, and this is just right, and then Morgan and maybe Dostoevsky, right? And this is important as a way to teach about rules, norms, how colleagues build on one another's work across generations. Of course. Next slide, please. But what we miss there in just the vertical histories are all of the social relations that they were embedded in, that both Nathaniel and Stephen spoke to. Next slide, please. So that slide of Morgan, and it's actually published in Nathaniel's book, is taken in the middle at Cold Spring Harbor. In 1915, the Scientific Research Board of the Eugenic Records Office. I don't mean this as a kind of indictment around Morgan, because I think there's been careful work that's shown how we would have to qualify that. The point is he's enmeshed in a set of relationships. Alexander Graham Bell, the industrialist inventor who has his own kind of points of entry in this around disability and deafness, is the chair of the Eugenics Congress that we heard about in 1921. Irving Fisher, who's an economist who teaches at Yale, at the same time he's with Morgan, is taking all of that information about genetics and basic Mendelian ideas and building a program, a nativist program around immigrant exclusion, around sterilization, et cetera. So they're enmeshed in a set of relationships. Next slide, please. And there's a wider sense. There's sometimes we say, they just didn't know any better. They're simply people of their time, as if there was a consensus among everyone that human hierarchies were natural and inevitable, and some people's, there was differential value assigned to life. But as this Congress is happening in 1921, right, this is the time of the Harlem Renaissance. The NAACP is founded. There's work against Jim Crow and lynching. There's rent strikes. There's people arguing for nativist work. There's the rebirth of Ku Klux Klan. All of these social relations are shaping the questions they ask. That's partly why it's so difficult to just take an individual and adjudicate guilty or not. It's a kind of empty exercise. And I think it's a disservice to students and to the clinical training we do when we don't embed these questions of discovery in these larger relationships. Next slide, please. Finally, just to wrap up, these are just a few examples. Fisher circulates in this world. He's at the International Eugenics Congress. They said, the science is sound. The problem is the public doesn't understand it. We need a concerted effort to educate the public that if we don't deal with the problem of degeneracy and race suicide, we're all done. So they bring to the New Haven Green the American Eugenics Society. Next slide, please. Madison Grant, also founder of the Bronx Zoo, Save the Redwoods League, has an environmental profile, the author of the leading white supremacist book of the 20th century, The Passing of the Great Race. He's in the mix too. And these are the, I think I have one more. Next slide, please. So from New Haven, where I work, this is what's disseminated in textbooks. These are examples of fairs. You can see on the right, this is the public's introduction to basic Mendelian ideas about inheritance. Alongside this contention, some people are born to be a burden on the rest. This was a kind of education slide, a light flashed once every 7 minutes, or sorry, once every 20 seconds when a low grade person was born, all the way up to once every 7 minutes when a high grade person is born. So it's right, those circuits of disseminating basic ideas are established early on. You can see the guinea pigs right there to the right. Next slide, please. So this the cover from this in eugenics, and you can see on the roots there that sense of all these disciplines have something to contribute to this project. So lest we think sober empirical scientific research was a fetter on this, it was the opposite. They understood all these disciplines having a role to play in this. And indeed, if you look through the program, that's what you see. Next slide, please. You can look through the program book, and I think, Stephen, you have copies of this. You will see papers reproducing norms that are deeply familiar. They're a part of the genealogy of work that's done in labs everywhere. And right next to it, next slide, deeply, cross sections, illustrations of brains alleging criminal and non-criminal, or, that there's chromosomes by race. The point is we have to look at the kind of energy and exchanges in between. We cannot just isolate and tell a genealogy of discovery alone. Okay, last couple, next please. So this is in the program book as well. This is in the program book. It's a call to end all immigration from Europe, especially Southern and Eastern Europe, decrying the alien presence. It's at the same scientific conference. So there is no easy isolation. Next, please. I just want to, again, see the ways that it kind of doesn't matter if just the bounds put by the discoverers on the work, because Whitney, who writes a book, this is a person at Yale, called The Case for Sterilization, is taking work by Mendel on the road to say the science supports why we need to sterilize people en masse. And indeed, 60,000 people are involuntarily sterilized through eugenic programs across the 20th century. Next slide, please. Again, examples of how they're trying to frame questions of inheritance, difference, and hierarchy, mobilizing scientific knowledge to reproduce and legitimate. Next one, please. The last two, they sponsor fitter family contests. So just like you could enter your livestock at the county fair and get a ribbon. And I tell my colleagues, all of this came from New Haven. This was all our people who did this. And they sent out displays. And in our archives, we see, will you give us a free space at the state fair so that we can show this and sponsor the contest? Next slide, please. Teaching genetics and eugenics side by side. There's very few genetics textbooks through the 1920s that don't have some significant section around eugenics. And this was, again, an instrumental strategic effort to do this. Next slide. This is just the last one. This is a illustration that a student made of the different figures in eugenics at Yale in the 20s and 30s. It shows people in psychology, anthropology, environment, forestry, the president, geography, et cetera, interacting. And so I just want to make that as a potential kind of like place to think from about that our disciplines in their minds were never isolated. They were always drawing ideas from one another for these purposes, and that the way we teach about it, I think, has to keep that in mind. Thanks.
Okay, let's get this forward. Okay. Hi, everyone. I'm excited to talk, and thank you both for inviting me to be here. Sad going third after all the great info's taken, but I just want to start with talking about how much of the conversation has been focused on the East Coast, but there was in fact a very active eugenics movement on the West Coast. And I think in the West Coast, it was basically rebranded as this kind of progressivism, but it was still here. And in fact, a lot of the leading scholars were actually recruited to the West Coast by the president of Stanford, David Starr Jordan. And I don't know how many people have seen in the news how people are calling for renaming of buildings. The same thing happened at USC as well with von Kleidschmied. Also our national parks, John Muir. So Madison Grant has a ton of stuff named after him as well. So this is not isolated to the East Coast only. This was a very big problem on the West Coast as well. And I specifically want to talk about one man, Lewis Terman, who David Starr Jordan brought over to Stanford. So Lewis Terman effectively made the IQ test into something. So Terman believed basically that like you cannot make someone smarter. It's just kind of like the genetics, the false science that was in hereditarianism kind of made him believe that the best thing we can do is categorize these people onto some level, and that's how we're going to kind of deal with it. There's no real way of fixing this. And actually, the original creator of the IQ test didn't even want to make it, Binet. And when he made this test, he kind of knew what was going to happen with it. So this is in France, but he was commissioned to do this, I think, by the governor. but he wanted to use his IQ test as a way to say, you know, this child needs to learn more. Let's identify where the issue is. Let's teach them. So this was a very mutable thing. For Terman, it was not, in fact, a mutable thing. So when Terman came to the West Coast, he eventually made his way to Whittier. So if you're from LA, Whittier is like, LA is weird shaped, but it's like one of the last places in LA, and then you move into OC. Well, in Whittier, we get the second location of the Eugenics Records Office that's been mentioned before. And Davenport actually said, okay, this is a great place to have it. And then Lewis Terman and his, I believe it was his graduate student, went out and they did mass IQ testing on children that were in homes in Whittier and Ventura, boys and girls that were in Whittier and Ventura. And then Yerkes, who was actually on your previous slide, did this mass testing in the military. They were alpha and beta tests. We also saw it later on in police departments as well with Volmer. So these IQ tests were used far and wide, and they were used for very, I would say, insidious purposes, especially in California, because then these children could effectively, like these juveniles, could be moved into state homes and then sterilized. So I think that that's something that many people do not know. I definitely did not know. And it turns out that California was actually a state that led sterilization. So California was responsible for roughly 1/3 of the forcible sterilizations in the US. So 20,000 sterilizations. And I think that's something uncomfortable. That's something that we have to sit with. And what really interested me in this, and I think gets me thinking about how can I make this a better place for people like me? So the majority of the victims of these forcible sterilizations would have been members of my family. And in fact, USC has this very sordid history where there were forcible sterilizations going on in Boyle Heights until very recent times, around the 70s. So some of these women are still alive. And there's actually a PBS documentary that tells their stories. So I think my introduction into eugenics was purely for thinking about, well, how do we learn about our field? How do we make our field a better place? How do we ask these questions about where is my work going to be in the future? And thinking critically about our work. So the majority of the time I'm thinking about, well, how can we make sure that we're not led astray by this flawed science? Because it's very easy to think vertically, extremely easy to think vertically. And we always have to be conscious about the fact that we can't just model environmental noise as a normal distribution, right? Like that's not what's happening. We have to think about socioeconomic status, what's going on. We have to think about the communities that we're working with. The reason that maybe we don't have diverse populations in our study is very well deserved. There should be reticence from people who were used for horrible experiments, right? Like we have the Tuskegee syphilis study that happened. We had horrible things that happened with the Havasupai where data was misused. And many of us tell these stories, but we don't actually think critically about why there might be a general reticence to not willingly give up data and participate. So that is my 5 minutes. Thank you.
Thank you.
Can everyone hear me?
Yes.
Yes. Okay, great. Thank you so much to the organizers for inviting me. And I wanted to start off by saying that definitely eugenics policies of the past are still affecting vulnerable populations today. Specifically, today I'll talk about the influences on Native Americans. For many years, or federal colonial policies that originated in what we now know as the United States over 300 years ago are still prominent and affecting Native communities today. These elimination policies were implemented in a variety of ways at different times throughout our history, through ethnic cleansing, forced removal, sometimes through massacres, and now in the 19th century through cultural assimilation. And often these policies were implemented using evolutionary theory and scientific racism as guiding principles. And these became officially instituted towards solving the Indian problem. Today, I'll talk about one prime example is the continued use of blood quantum in classifying who is Native. the notion of blood quantum was created to really track racial ancestry and define legal rights. It was thought that these genetic population categories could be objectively determined and quantified by their blood. So the idea of racial blood was aided by Francis Galton, who we heard about, and was a justification for Black slavery, as well as dispossession of land from Native peoples. It facilitated the displacement of and dispossession of Native identity as well. Today, blood quantum is still a mechanism of forced assimilation, which is aided by scientific racism for legally defining a Native American. So we can show my slide. Today, I'm showing this image today. This is actually my certificate of Indian blood, or CIB. Native Americans and American Indians are the only racial ethnic group who have to prove that we are who we say we are. We are not only members of a cultural and ethnic group, but we are also citizens of our tribal nation. So this is a piece of paper that was given to my parents when I was born, designating what my blood quantum is. So the legal significance of blood quantum is currently used to determine eligibility for federal resources and services. It's used to determine tribal membership, and it's used to delineate economic and political benefits. Blood quantum has separated thousands of people from their Native American ethnic heritage arbitrarily by defining who and who is not a person of Native American ancestry. For some individuals, blood quantum is a eugenics policy designed to statistically exterminate the remaining Native American people. And this is because you can never increase your blood quantum, you can only decrease it. So it definitely has implications for who tribal members marry and the future of the tribe. So we can take down the slide now, but there are other ways that eugenics policies have played a significant role in Native communities. And this includes how federal governments and other authorities have long tried to control Indigenous families and women's reproduction through forced sterilization, as well as removal of Indigenous children from their homes into foster care systems. In the 1970s, doctors in the US estimated that 25 to 42% of Native American women of childbearing age were sterilized. And in 1976, the government admitted to this. In a study, it was estimated that over 3,400 American Indian women between 1973 and 1976 were sterilized. 2 years earlier, another study by Dr. Connie Pinkerton found that 1 in 4 American Indian women had been sterilized without her consent. And in this research, it showed that the Indian Health Services, where many Native people get their health care, they really singled out full-blooded Indian women for sterilization procedures. And it's estimated that the percent of American Indian women who were sterilized might even approach 50%. So this was only in the 70s. So it was my parents, my aunties, and my family who were part of this sterilization effort. Lastly, racial politics rooted in eugenics affected the erasure of Native peoples and communities. This refers to the removal and erasure of Native peoples from official records. So the use of documents to affect the erasure of Native identity through manipulation of racialized descriptors and also altering documents themselves. So society refused to recognize Natives, especially in the southeastern United States, as anything other than Black. And these were targets of eugenicist policies. It made it illegal. Some states made it illegal for people to identify as American Indians in the early 1900s. So racialization equals detribalizations. So in closing, eugenics is not in the distant past, but it still affects, the effects are still present and affecting people now. Thanks.